Hello friends, welcome back to our YouTube channel Doctor's Corner. In this today's video, we are going to discuss about the skeletal muscles. Skeletal muscles. So as you might be knowing that 40% of our body mass or weight is mainly made up of the skeletal muscles, whereas 10% is made up of the cardiac muscles and smooth muscles together. Okay, so in this particular video we are going to discuss about the anatomy and physiology of the skeletal muscles. Anatomy and physiology of the skeletal muscles. More specifically, we will be discussing the functional anatomy in this particular lecture. Okay, so consider this as a skeletal muscle. So I am just drawing a rough skeletal muscle. Consider this as a skeletal muscle. Okay, so you might have seen the skeletal muscle in the dissection during the first year MBBS, various skeletal muscles in the upper limb and the lower limb. So more than 600 skeletal muscles are present in our human body. So if we take a cut section of this skeletal muscle, how does it look? Okay, how does it look? So there is a outer covering of connective tissue is present. That outer covering of on the muscle belly, the connective tissue which is covering on the outer aspect, it is known as epimysium. Epimysium. Okay, and inside this muscle belly, it is present that muscle fibers are present in the form of bundles. These bundles are known as fascicles or fasciculus. Okay, so they are present in the form of fascicles or fascicules. So these fascicles are again surrounded by a connective tissue sheath that is known as perimysium. Perimysium, right? Now inside this perimysium comes the muscle cell. Now as you know the basics, that the tissues are mainly made up of cells, that is the cells, group of cells having a similar function are known as tissues. So the muscle cells, that is this muscle, skeletal muscle tissue is made up of muscle cells, and that muscle cells are known as muscle fibers. So they are present in the form of a cylindrical shaped muscle fiber. So these are the cylindrical cells which are present in the form of bundles, that is the fascicles. Okay, so each of this muscle fiber will be present in the form of the bundle. So this is the arrangement of the muscle in our human body. Okay, so if you see a cut section of the muscle, you will find this type of the structure. Okay, so firstly the outer covering, which is known as epimysium, then these fascicles are covered by connective tissue which is known as perimysium, right? So again this single muscle fiber, which is a muscle cell, is further covered by one more connective tissue sheath. That connective tissue, it is called as endomysium. Okay, endomysium. So all these connective tissue sheets, that is the epimysium, endomysium and perimysiums, are interconnected to each other and helps in the smooth contraction of these skeletal muscles. Okay, we'll discuss about that in detail later in the next video about the molecular basis of the muscle contraction. Right now we are concentrating on the functional anatomy. So now we'll take one skeletal muscle fiber. Okay, one skeletal muscle fiber, how does it look, and what exactly it is present inside this muscle cell or the muscle fiber. Okay, so the outer membrane, that is the cell membrane of the muscle cell, it is called as sarcolemma. Okay, so sarcolemma basically consists of the plasma membrane of the skeletal muscle as well as one thin layer of the polysaccharide. Together it is known as sarcolemma. Okay, now as any other cell, it has the cellular constituents like nucleus. So it is a multinucleated cell, and typically the nucleus is present on the peripherally located, and it has the other organelles like endoplasmic reticulum, which is known as sarcoplasmic reticulum. Again we'll discuss about that in the next lecture, in the sarcotubular system. Okay, then mitochondria. But one of the very important component here is in the skeletal muscle cells is myofibrils. Okay, the presence of this very thin micro structures that is known as that is known as myofibrils. Okay, so this myofibrils, there are many myofibrils will be present in each of the muscle fiber. Okay, so this myofibrils, it may range from 100 to thousands in a one single muscle cell or the muscle fiber. So if we enlarge and see one single myofibril, okay, if we enlarge and see one single myofibril, so where this myofibril is present? Myofibril is present inside the muscle cell. Muscle cell is nothing, a muscle fiber. Okay, so we have already discussed how exactly the arrangement of the muscle cell. So in the myofibril, these myofibrils, if we see, so the appearance will be having a typical alternate light and dark bands. Okay, there will be dark bands and there will be light bands. Okay, so typically alternatively you will find in one myofibrils, if you see, so this there are typical arrangement of the light and the dark bands. Okay, so this dark band, it is typically, it is known as A band. Okay, it is known as A band because it is anisotropic to the polarized light, whereas this light band, it is known as I band. So typically there will be alternate arrangements of the light and the dark bands in the myofibril. So because of the presence of this light and the dark band, this muscles appears as the striated muscles, that is there are striation scenes. Even in the light microscopy you can see the striations, and these striations of the skeletal muscle is because of this myofibrils. Okay, now in this myofibrils, this dark band or the light band, what proteins they are made up of? What proteins? We'll discuss about that proteins. So mainly they are made up of this following proteins. So firstly, the most important one are the contractile proteins, which will help in the muscle contraction. Okay, we'll discuss about this, how exactly the muscle will contract in the molecular basis of the muscle contraction. Time being in the structure, just concentrate what proteins are available. So the main two important contractile proteins are actin and myosin. Okay, actin and myosin. So we'll discuss about these two structures of these two important proteins. Apart from this contractile protein, there are other proteins also which are present in the myofibrils, and these are known as myofilaments. Okay, these proteins are arranged in the form of the thick and thin filaments. We'll discuss now. Then there is something known as regulatory proteins, which will regulate this contractile proteins. So the regulatory proteins which are present in this myofilament or along with the myofilament in this myofibrils are tropomyosin and troponin. Okay, tropomyosin and troponin. Then finally also there are something known as anchoring proteins. Okay, there are anchoring proteins present. So the anchoring proteins which are present in the myofibrils are titin. We'll discuss about this, where exactly they are present and how they look, or the structure also if possible. Nebulin, then alpha actinin and dystrophin. Okay, there may be other proteins also, but the important proteins we are discussing. So basically it has the contractile proteins, regulatory proteins and the anchoring proteins. Okay, now these proteins, how they are present, or how all these proteins are located in this myofibril. Okay, so basically if we further enlarge and see in an electron microscope, how does this look? So first we'll come to the A band. Okay, so this A band is mainly made up of thick filaments. It is made up of thick filaments. So that's why it appears darker. So thick filaments will be present like this. Okay, A bands are mainly made up of thick filaments like this. So that's why they appear a bit darker. So thick filaments. So they are made up of thick filaments. Okay. So now these thick filaments mainly consist of this protein myosin. Okay, this protein myosin. Okay, the contractile protein myosin. Now what is the structure of the myosin? It is a heavy protein having a molecular weight of around 4,80,000. Okay, having a molecular weight of around 4,80,000. So it consists of the tail portion. Okay, so its tail portion, double helix like this, then a neck, then a globular two heads will be there. So it has two heavy chains, okay, two heavy chains, each of weighing around 2 lakh molecular weight, and 4 light chains weighing around 20,000 molecular weights. 4 light chains and 2 heavy chains, okay. So this is, it has, and more specifically it is the myosin 2. It is known as myosin 2. It has 2 heads, myosin 2, and one of the activity of this, functional activity of this myosin is it has an ATPase enzyme activity, that is it can convert ATP into ADP and phosphate, okay, which we'll discuss in the molecular base of muscle contraction. So timing, remember this one. So this thick filaments is mainly made up of this myosin molecules. Now these myosin molecules, the arrangement of these myosin molecules, typically the head portions, okay, head portions are present at the periphery, and whereas the tail portions are situated in the center. So because of this typical arrangement, that is this head portions are at the periphery and this tail portion is at the center, so in the center it will appear a bit thinner, though it is a dark filament, that the center region will be thinner, appears lighter as compared to this peripheral. So this thinner portion, it is known as H zone, okay, it is known as H zone, whereas this peripheral portion together, this totally it constitutes of A band. So A band mainly consists of myosin filaments or the myosin proteins, okay. So these myosin proteins are arranged like this, and they will appear darker, like this, and in the center there is a lighter zone that is known as H zone, okay, H zone. Again, further in the H zone there is one line, one darker line it is seen, one straight darker line is seen, that is known as M line, okay, that is known as M line. It consists of the proteins known as myosin and C protein and creatine kinase, okay, so M line. Now on either side of this dark band there is the light band, also known as I band. So this I band mainly consists of the actin filaments and the other proteins filaments. What are that protein filaments we'll discuss. So it is mainly made up of actin and other. So with the help of this blue color lines what I have drawn, it is the I band, that is the light band, I band, okay. Then again further it will continue as A band, so alternate A and I bands will be there. So I band is mainly made up of the thin filaments, The thin filaments are nothing but actin, okay, actin. So in this thick filament around 1500 myosin molecules will be present, okay, in a one myofibril around 1500 myosin molecules will be present, whereas around 3000 actin molecules will be present, okay. So this actin filaments are generally, if it is a single one, then it is called as G actin. It acts as a monomer. When this single monomer polymerize to form a chain, then it is called as F actin. So in the skeletal muscle, so actin is present in the form of F actin, okay, in the form of F actin polymerize. So around 3000 such molecules will be present in a myofibril. Then not only actin, so this thin filament is mainly made up of actin molecules, not only actin, but it is also consists of two important proteins, mainly actin, then tropomyosin and troponin. So thin filament is made up of actin, tropomyosin and troponin, and thick by the myosin. So where exactly this tropomyosin and troponin molecules are present on this thin filament? So there is, in the groove there is a ribbon like one protein, okay, ribbon like, thread like or ribbon like protein will be there, a rope like structure which is present in between the groove of this F actin. With the help of this green color I have just demonstrated you. So this, it is called as, this protein, it is called as tropomyosin. So this tropomyosin will cover the active sites of the actin. Generally there is an ADP activity that is called as the active site of the actin, on which the myosin molecule can bind, okay. Now apart from this there are other proteins known as troponin, okay. Troponin molecule will be present that is present on the tropomyosin and actin molecule on the thin filament. So this troponin molecule has the 3 subunits: troponin C, troponin T, and troponin I. So troponin C is mainly for the binding of the calcium ions, troponin T is mainly for the binding of the tropomyosin molecule, and troponin I will bind to the actin molecule. So the basic function of this troponin molecule is to bind the tropomyosin molecule with the actin molecule. Okay, now what is the functional significance is during the process of muscle contraction, the calcium ions bind to this troponin C subunit. So when the calcium ion binds to the troponin C subunit of this troponin molecule, it undergoes some conformational changes that will pull the tropomyosin molecule out from the actin. That is, the active sites of the actin now will become exposed for the muscle contraction. Later we will discuss in detail in the molecular basis of muscle contraction. Time being, just remember this one. So what we have discussed till now, that this myofibril which is present in the muscle cell. So muscle cell have numerous myofibrils. So myofibrils have alternate light and dark band. Dark band is known as A band, and in that A band mainly myosin proteins or myofilaments are present, whereas in the I band actin, tropomyosin and troponin are present. Right, now in this I band, you can see there is one disc or one line seen. That disc or one line, it is known as Z disc or Z line. Okay, Z disc or Z line, and it is made up of one protein. So chemically it is made up of one protein. The protein which form this is the alpha actinin. So in the Z disc there is one protein, that is alpha actinin. Okay, so this Z disc is present in the I band. Now the distance between these two Z disc in a myofibril, it is called as sarcomere. Sarcomere. So what is a sarcomere? By definition, it is the functional unit of the skeletal muscle. Structural and functional unit of the skeletal muscle is known as sarcomere, and typically it will extend from the two Z lines, in between two Z lines. So typically it's on an average the length will around 2.5 microns. Okay, so how 2.5 microns? So this A band will have around length of around 1.6 micrometer, and this I band typically have 1 micrometer length. Okay, so basically one sarcomere will have half of the I band and one full A band, and again half of the I band. So this will form one sarcomere, and it will be around 2.5 microns. Okay, so during the muscle contraction this will be reduced. The size of this sarcomere is reduced, that is, it will be around 2 microns, and during the muscle relaxation it will increase. It may extend up to 3 microns. Okay, so this is all about the sarcomere and the light and the dark bands and the thick filaments and the thin filaments. Now there are certain anchoring proteins like titin, nebulin and dystrophin. So where exactly this proteins are present in a myofibril? So in the myofibril, this protein titin, okay, titin is one of the largest protein which is present in the human body. So generally it will connect this Z line, okay, or the Z disc or the Z disc with this M line in the center. Okay, with the M line in the center, with the help of this green pen I have shown this. So it is an elastic protein which will connect the Z line with the M line, and it is a very elastic and a larger molecular weight protein. Okay, so it will help in the process of muscle contraction. It acts as a series elastic component. Later we will discuss, when we will discuss the isotonic and isometric contraction in the properties of the skeletal muscle. Timing, just remember this titin is present in this and will connect the Z line and to the M line. It is a very elastic protein. Okay, then we have one more protein known as nebulin. Okay, so in the anchoring proteins we have discussed about the alpha actinin and titin. Then nebulin, this particular protein nebulin is present as a ruler or marker over the thin filaments. Okay, so as you know, the thin filament is made up of this actin, right? So throughout this I band it will act as a marker or a ruler. This is called as nebulin protein. Okay, so nebulin protein will be present in the thin filament. Okay, they will be present in the thin filament nebulin. Then one more is alpha dystrophin or just dystrophin. So this particular protein dystrophin. So this dystrophin molecules will be connecting this myofilaments or the myofibrils to the sarcomere, okay, that is the outer covering. So this, it is known as dystrophin. There is a clinical significance of this molecule dystrophin. So whenever this particular protein dystrophin is not properly formed or absent, then it leads to the condition known as muscular dystrophies, okay, muscular dystrophies, like example Duchenne muscular dystrophy, Becker muscular dystrophy. We'll discuss in the pediatric section or in some other section regarding this muscular dystrophies later. Timing, just remember, this dystrophin molecule will connect this myofibrils to the sarcolemma, and if this is absent, then the whole cytoskeleton, that is the structure, will not remain same, and it leads to the destruction or fibrosis of the skeletal muscles, okay. So this is all about the skeletal muscle. So just to summarize, so skeletal muscle structure. So today's topic is structure of the skeletal muscle. So in this, what we have discussed is how the arrangement of the muscle cells in a skeletal muscle, how they are arranged in the form of the muscle fibers in the bundle known as fascicles, and later each muscle fiber consists of mainly this myofibrils and other cellular organelles, and this myofibrils has the alternate light and the dark band, which is the cause of the striations in the skeletal muscle, and this is mainly made up of this following proteins like actin, myosin. We have discussed how exactly these proteins are present. So in the darker band or the A band, myosin molecules are present. We have discussed the structure of myosin also, and in the light band, actin, tropomyosin and troponin molecule is present. Then we have discussed about the Z line or the Z disc or the Z disc and the sarcomere, and also the regulatory proteins like nebulin, actinin and titin and alpha dystrophin, okay. So that's all in this today's lecture. Thank you.